Now that we are done covering the metabolism of carbohydrates, it's time to consider the metabolism of another macronutrient, fats. As we did for glucose and other carbohydrates, I want to begin with a quick overview of the pathways we will cover to get some intuition on this nutrient. In a similar fashion to glucose, fat metabolism is composed of anabolic and catabolic reactions. Also, one good aspect about the metabolism of all nutrients is that they all end up converging on the same few metabolites. As such, we will keep our block of reactions for glucose and add the reactions related to fat metabolism. So, the majority of the fat that we get from our diet comes in the form of triacylglycerols. As it says in the name, this molecule is made out of a glycerol and three acyl groups, also known as fatty acid chains. The fatty acid chains, which are abbreviated by capital R on the metabolic map, can vary in both length and bonding, so in comparison to the carbohydrates we have seen, fat molecules are way more diverse. Right after this overview, we will cover the terminology for all these different types of fatty acids. Now, the energy of triacylglycerols is stored in the fatty acid chains. As such, the first reaction pathway to consider is how triacylglycerols are broken down into their fatty acid chains. These fatty acid chains can then be degraded in a process called beta-oxidation, which will produce acetyl-CoA, NADH, and FADH2. All these products can then be used by the citric acid cycle and the electron transport chain to make energy. Importantly, beta-oxidation happens in the mitochondrion, so we will have to consider how fatty acids are transported into the cell. As a whole, this is how energy is made from fatty acids. If glucose is not available to the body, some cells in the body can turn the acetyl-CoA made by fatty acid degradation into ketone bodies. These ketone bodies can then be reversibly turned again to acetyl-CoA and fuel the cells that primarily function on glucose, for example neurons. When we consider the anabolic side of fats, fatty acids can be reformed from acetyl-CoA with an input of energy in ATP and NADPH. This reaction is pretty analogous to gluconeogenesis, where pyruvate and some energy is used to remake glucose. The newly made fatty acids can then be stored back into triacylglycerols in a process that requires ATP to happen. This step is analogous to glucose getting stored in glycogen. In all, the extent of the reactions that we will cover look like this on our metabolic map. Now, just like we've seen with the metabolism of carbohydrates, the metabolism of fats is also subject to hormonal control, as you can see up top. Accordingly, we will see how the different pathways are regulated by insulin, epinephrine, and glucagon, but as I mentioned during our discussion on carbohydrates, the details that concern the organs and different types of tissues that can be affected by the hormones will be covered in a later section. Also, in this video, we will not cover how fats are transported across the body because I want to make a video on the bloodstream that deals with this aspect separately. So, to cover the metabolism of fats, we will first go through the catabolic reactions from triacylglycerols to acetyl-CoA and then make our way back with the anabolic reactions. But before we do so, I want to take a look at some terminology which will be useful throughout our discussion. So, first of all, fats are also known as lipids, which is a word that encompasses fatty acids, triacylglycerol, steroids, and basically any organic compound that is not soluble in water but is in a nonpolar solvent. Now, if we consider the structure of a triglyceride, as I mentioned previously, it is made out of a glycerol backbone and three fatty acid chains. The glycerol backbone will always be the same, but you will notice that the fatty acid chains can be quite different from each other in terms of length and bonds. The length of fatty acids usually varies between 14 and 20 carbons, but the bonding is where fatty acids differ the most. The two general types of fatty acids that can be differentiated based on bonding are the saturated and unsaturated fatty acids. To put it simply, saturated fatty acids only contain single bonds between each carbon of their tail, whereas unsaturated fatty acids have at least one double bond. As an example for saturated ones, we can consider palmitic acid, which is a 16-carbon long fatty acid. For unsaturated ones, we can consider arachidonic acid, which has 20 carbons and contains many double bonds. When an unsaturated fatty acid has only one double bond, it is referred to as monounsaturated, and when it has two or more, it is referred to as polyunsaturated. In this instance, arachidonic acid is a polyunsaturated fatty acid. Now, an important implication of having unsaturations is the fact that the double bond can be in a cis or a trans conformation. If we consider the hydrogens that are typically hidden in my molecules, cis conformations occur when hydrogens are on the same side, and trans conformations when they are on opposite sides. Accordingly, the carbon groups will be on the same side in a cis conformation and on opposite sides in a trans conformation. This aspect directly influences the shape of fatty acids. For example, if we consider lactic acid and oleic acid, they are both monounsaturated fatty acids with 18 carbons and a double bond occurring at the 9th carbon, but lactic acid is trans and oleic acid is cis. Due to the carbons being on the same side, oleic acid has this bend in its structure that lactic acid does not have. This change in shape can influence the properties of cell membranes and can even be a health risk factor, as trans fats can increase the chances for cardiovascular diseases. Lastly, as a quick note for arachidonic acid, I showed its structure in this linear way to make space, but in cells, the structure looks more like this due to every bond being cis and thus making this bend in the structure. Alright, now that we have this in our toolbox, let's discuss the catabolism of fatty acids, starting with triacylglycerols. The process of triacylglycerol breakdown is also known as lipolysis, and it pretty much has two ways of occurring depending on where we are in the body. In the liver and adipose tissues, which are basically just cells that have the sole purpose of storing fat, the two sites express adipose triglyceride lipase, hormone-sensitive lipase, and monoglyceride lipase. Each of these enzymes have the function to remove a fatty acid. The only difference is the initial substrate that they do it from, so ATGL cuts one fatty acid from triglycerol, HSL cuts one fatty acid from diacylglycerol, and so on. Through this pathway, glycerol ends up being produced. The other way to undergo lipolysis happens with lipoprotein lipase and pancreatic lipase. These two enzymes directly convert triacylglycerols to monoacylglycerols and thus liberate two free fatty acids. Pancreatic lipase is expressed in the gastrointestinal tract and basically allows the body to ingest fat from food during digestion. On the other hand, lipoprotein lipase is expressed in the bloodstream, and the lipolysis that it performs generates fatty acids for muscles and adipose tissues to absorb. Now, the reason why adipose tissues and the liver have these three extra enzymes to degrade triacylglycerols is because they are the tissues that deal the most with fat. Adipose tissues are the main source of fat storage, and the liver plays an important role in managing fat across the body. Accordingly, lipolysis, especially in adipose tissues, is subject to hormonal control. Since these hormones have a dual effect on catabolic and anabolic pathways, I will discuss the effects of glucagon, insulin, and epinephrine in a later section when we will cover the anabolic process that reforms triacylglycerols. Until then, let's take one of the fatty acids that we have made from lipolysis and let's see how it can generate us some energy with beta oxidation. As I mentioned in the overview, since beta oxidation happens in the mitochondrion, we have to first consider how it's transported there. To make it happen, fatty acids first need to be charged with energy and be converted to acyl-CoA, which is simply our fatty acid with coenzyme A. Coenzyme A, or simply CoA, is a molecule that we have seen during our discussion on glucose, and if you recall its structure, you might remember that what's important about it is this bond highlighted in red between the fatty acid and the S. Indeed, this bond is important because its cleavage releases a lot of energy, and this energy is typically harnessed to facilitate downstream reactions. A clear-cut example was in the citric acid cycle, where in the first reaction, the release of CoA from acetyl-CoA provides the energy to make citrate. Now, the addition of a CoA to fatty acids happens in two steps and is catalyzed by acyl-CoA synthetase. In the first step, ATP is used to add an AMP moiety to the fatty acid, which will be replaced by CoA in the second step. The two phosphates that are released in the first step can then get cleaved by the enzyme inorganic pyrophosphatase, and this process releases the energy that will supply the formation of acyl-CoA. When acyl-CoA is made, the process of transporting it into the mitochondria can occur via a Carnitine shuttle. If we consider a chunk of the inner mitochondrial membrane on the metabolic map, the inner membrane contains a transporter called the carnitine carrier protein, which is able to move around compounds that have carnitine across the membrane. As such, With the acyl-CoA that we have made, the first step of this shuttle is to replace the CoA with carnitine, which produces acylcarnitine and will be able to be transported across the membrane. This reaction gets catalyzed by carnitine palmitoyl transferase 1, and you will notice that this is a membrane protein that is located on the outer side of the outer mitochondrial membrane. Inside the mitochondria, the enzyme carnitine palmitoyl transferase 2 can essentially do the reverse and remake acyl-CoA that will be able to get processed by beta oxidation. The carnitine can get released and travel back out of the mitochondrion where it can help other fatty acids get in. You will notice that in comparison to CPT1, CPT2 is located on the inner portion of the inner mitochondrial membrane. In all, CPT1, CPT2, and the carnitine carrier protein allow an efficient mechanism to send fatty acids into the mitochondrion by shuttling carnitine back and forth. You can see throughout the pathway that the reaction catalyzed by CPT1 is in violet, whereas all the others are in gray. The reasoning for this is because this reaction is the rate-limiting step for this entire process. As such, this step constitutes the perfect place to regulate the entry of fatty acids in the mitochondrion for degradation. It turns out that the main mechanism to regulate the entry of acyl-CoA is through the inhibition of CPT1 by malonyl-CoA. As we will see later, malonyl-CoA is the starting point for fatty acid synthesis, and fatty acid synthesis generally happens under conditions of high energy. Thus, since the cell has a ton of energy, it makes sense that the accumulation of malonyl-CoA blocks the degradation of fatty acids since it does not want the fatty acids to be wasted away. With this being said, now that our fatty acids are inside the mitochondrion as acyl-CoA, let's cover how beta oxidation makes energy. Given the wide diversity of fatty acids, whether they are saturated, monounsaturated, or polyunsaturated, beta oxidation has to be flexible enough to accommodate the degradation of all these variants. As such, while we go through this process, we will consider a few scenarios to understand how various fatty acids get degraded. To start, we will consider the most simple case, a 12-carbon saturated fatty acid. Due to their long hydrocarbon tail, the degradation process of fatty acids does not happen in one set of reactions. Instead, beta oxidation is set up as a cyclic set of reactions that will degrade 2 carbons every round. The 2 carbons that are removed at each round will always come from the end that has the CoA. As such, let's consider the first 3 carbons, which on the entire molecule correspond to what's boxed in red. The capital R represents the rest of the molecule, and this will be the starting point for saturated fatty acids. Before we get into the reactions, there are 2 bits of terminology that I want to review to properly understand what beta oxidation is. The first aspect comes from organic chemistry, and it is the notation of carbons. If we consider our acyl-CoA, the first carbon away from the functional group, here highlighted in blue, is denoted the alpha carbon, and the second carbon away is denoted the beta carbon. As we will see shortly, the degradation of fatty acids happens by breaking the bond between the alpha and beta carbons. The other aspect of terminology is to recall what a reduction and an oxidation is in the context of a metabolic pathway. To put it simply, oxidations are a loss of electrons and reductions are a gain of electrons. Since matter is conserved, reductions are always coupled to oxidations. Generally speaking, oxidations will make substrates less complex and lead to their degradation, whereas reductions make substrates more complex. The first reaction in this entire process, which is catalyzed by acyl-CoA dehydrogenase, illustrates pretty well this concept. Basically, the enzyme oxidizes acyl-CoA by removing electrons, which turns the bond between the alpha and beta carbons into a double bond. The electrons that are removed from this bond are used to reduce FAD to FADH2. Because of the double bond, the acyl-CoA is now called trans-delta-2 enoyl-CoA. Here, the delta-2 simply signifies that the double bond, which is trans, is on the second carbon. Moving forward in the pathway, the second reaction is mediated by enoyl-CoA hydratase, which uses water to reform a single bond and add an alcohol on the beta carbon. This produces 3L-hydroxyacyl-CoA. The enzyme 3L-hydroxyacyl-CoA dehydrogenase then uses NAD+ to oxidize the alcohol group on the beta carbon and turn it into a ketone group. This produces 3-ketoacyl-CoA and 1 NADH. In the final step of the cycle, The enzyme thiolase cleaves the bond between the alpha and beta carbon, which produces one acetyl-CoA. The enzyme also takes one CoA and adds it to the beta carbon and reforms our acyl-CoA, but now with two less carbons. Based on the initial molecule, the alpha carbon is now in the acetyl-CoA and the beta carbon stays in the 10-carbon acyl-CoA. Hopefully by now you can get an idea why this process is called beta oxidation. Now, after one round of beta oxidation, our 12-carbon acyl-CoA is shortened into a 10-carbon acyl-CoA, and this generated one molecule of acetyl-CoA, one molecule of NADH, and one molecule of FADH2. In the following rounds of beta oxidation, the degradation will again be carried out at the first three carbons. Since the same reactions will take place, we can predict that after the second round, another acetyl-CoA, NADH, and FADH2 will be made. To completely degrade our 12-carbon molecule, it will require 3 additional rounds of beta oxidation, which in total will generate 6 acetyl-CoA, 5 NADH, and 5 FADH2s. After covering all the scenarios, we will go back to these products to understand what happens with them. Now, in the second scenario, let's consider the degradation of a saturated fatty acid that has an odd amount of carbons, for example, 11. For the most part, the fatty acids that we consume and are produced in our body will have an even amount of carbons because the synthesis is performed in rounds of 2 carbon additions, but odd-chain fatty acids can be found in some fish and plants. So, to degrade these fatty acids, it will start the same way we have seen with the even chain, as 2 carbons get removed every single round. The process gets a little different once the fatty acid reaches 5 carbons. When the 5-carbon chain reaches the final step, since thiolase can only perform the cleavage between the alpha and beta carbons, this reaction will produce a 2-carbon acetyl-CoA and a 3-carbon molecule called propionyl-CoA. Fortunately, this propionyl-CoA is still useful as the cell can convert it to succinyl-CoA, which is a substrate of the citric acid cycle. To do so, propionyl-CoA gets a CO2 group added onto it by the enzyme propionyl-CoA carboxylase, and this produces methylmalonyl-CoA. As you can see, this reaction requires an input of ATP to happen. Then, the enzyme methylmalonyl-CoA mutase and epimerase convert the skeleton of methylmalonyl-CoA to make succinyl-CoA. In all, our 11-carbon saturated fatty acid generates 4 acetyl-CoAs, 1 succinyl-CoA, 4 NADH, and 4 FADH2. With the even and odd saturated chains covered, let's consider what happens when there is a double bond. With unsaturated fatty acids, there are basically 2 possibilities regardless of whether the fatty acid is mono or polyunsaturated. Either the double bond occurs on an odd carbon or it occurs on an even carbon. Let's begin with the odd carbon double bond by considering this 12-carbon monounsaturated fatty acid. Here, the double bond occurs between carbons 9 and 10. Now, since most of the chain is still saturated, the first few rounds of beta oxidation will occur just as we've covered. Each round, 2 carbons are removed from the fatty acid, and this generates 1 acetyl-CoA, 1 NADH, and 1 FADH2. The difference with odd unsaturations comes when we reach round 3, in which you can see our double bond is now between the 3rd and 4th carbon of the chain. If we consider this substrate as the starting point for double bonds at odd carbons, this compound is problematic for beta oxidation because it is not a substrate for enol-CoA hydratase. As such, it first needs to be processed before it can be oxidized. The solution for this problem is the enzyme enol-CoA isomerase, which converts the delta-3 enol-CoA to trans-delta-2 enol-CoA and consequently allows the rest of the chain to be processed. You will notice that the name does not mention whether the bond in delta-3 enol-CoA is cis or trans, and that is because enol-CoA isomerase is able to convert the compound into the trans-delta-2 version regardless of that. Accordingly, this allows both cis and trans fats to be processed by beta oxidation. Another important aspect in this is that since the chain already bears a double bond, it skips the acyl-CoA dehydrogenase step and thus skips the production of FADH2. Accordingly, in our final yield for this molecule, 1 less FADH2 molecule will be produced. The final scenario that can arise is the double bond being on an even carbon, for example this 12-carbon monounsaturated fatty acid with a double bond at the 8th carbon. To see the unique reactions of this fatty acid type, we need to consider what happens after we complete the second round, as we will get a fatty acid with a double bond on the fourth carbon. This fatty acid will be the starting point for double bonds at even carbons. Now, just like the other saturated chain, this molecule gets oxidized by acyl-CoA dehydrogenase, which produces 2,4-dienol-CoA. This 2,4-dienol-CoA is problematic for the rest of beta oxidation because it inhibits enol-CoA hydratase and thus blocks the oxidation of the fatty acid. So again, the cell needs to fix the fatty acid before it can process it. To do so, the enzyme 2,4-dienol-CoA reductase uses NADPH to basically add electrons into the compound and turn it into delta-3-enol-CoA. Just like we've seen with odd unsaturations, the delta-3-enol-CoA can be converted back into a substrate that enol-CoA hydratase can process. However, since the compound still gets processed by acyl-CoA dehydrogenase first, double bonds on even carbons still produce the FADH2. In total, this monounsaturated fatty acid produces the same yield as the saturated one. To see the complete picture of beta oxidation, let's summarize all the processing rules we have covered. For saturated fatty acids, if the chain is 2 N-carbons long, then beta oxidation will produce N amounts of acetyl-CoA and N-1 amount of NADH and FADH2. If we take scenario 1 as an example, N would be equal to 6. If the chain is odd, so 2N plus 1 carbons long, then the output will be N-1 acetyl-CoA, NADH and FADH2, and 1 succinyl-CoA. Also, don't forget that the conversion to succinyl-CoA costs 1 ATP. When it comes to the unsaturated fatty acids, as we've seen, what matters is on what carbon the double bond falls more than the chain length itself. If the double bond falls on an odd carbon, 1 FADH2 is subtracted per double bond. If the double bond falls on an even carbon, then the FADH2 is produced and it can basically be treated as the saturated fatty acid. You can keep in mind that it costs 1 NADPH per even processed double bond, but this should not impact the energy yield. These few rules should cover the majority of the biological fatty acids that get degraded in the body by this process. Now, when it comes to all the products of beta oxidation, these are all the substrates that can be used by the same pathways that make energy out of glucose. Acetyl-CoA and succinyl-CoA are both substrates of the citric acid cycle and allow the generation of even more NADH and FADH2. One turn of this cycle, which is fueled by 1 acetyl-CoA, produces 3 NADH, 1 FADH2, and 1 GTP. All the NADH and FADH2 molecules that are produced from the citric acid cycle or beta oxidation can then be used in the electron transport chain to generate energy directly. If you want more information on how the citric acid cycle and the electron transport chain work, you can consider going back to the section on glucose where we go into more detail in each of these pathways. But basically, the idea is that the oxidation of NADH and FADH2 into NAD and FAD is used to reduce these CoQ molecules, and this process of transporting electrons across the chain allows to pump protons in the intermembrane space of the mitochondrion. This gradient of protons can then be used by the protein ATP synthase and directly make ATP. Since this entire process requires the presence of oxygen, it is also known as oxidative phosphorylation. As we covered in our discussion on oxidative phosphorylation, 1 NADH is responsible for the production of about 2.5 ATPs, and 1 FADH2 is responsible for the production of 1.5 ATPs. These values allow us to determine the true energetic yield for fatty acids. Now, one important detail to add is that in comparison to NADH, FAD and by association FADH2 are not soluble. Usually, the FAD is contained within a protein of some sort. In the case of the acyl-CoA dehydrogenase reaction, the electron transport flavoprotein has the FAD and is responsible for delivering the electrons to CoQ. Energy-wise, this protein and complex 2 basically act the same way since they have the same end goal and neither of them pump any protons. Lastly, I want to mention that if figuring out the energy yield for a given fatty acid is important for you, keep in mind that some energy is spent to transport the fatty acid inside the mitochondrion. Since the ATP that is spent is degraded to AMP directly, some textbooks will consider that 2 ATPs are spent to compensate for the loss in 2 phosphates. If anything is unclear, don't hesitate to ask away in the comments. Alright, before we move on to the anabolic side of fats, I want to briefly discuss ketone bodies and their purpose since they indirectly tie back to beta oxidation. So, there are three ketone bodies: acetone, acetoacetate, and beta-hydroxybutyrate. Their production, which is also known as ketogenesis, begins by the condensation of two acetyl-CoA molecules to produce acetoacetyl-CoA. This reaction is mediated by thiolase, which, as you may remember, was the final enzyme in beta oxidation that was producing the acetyl-CoAs. So basically, ketogenesis starts with the inverse reaction of the last reaction in beta oxidation. The acetoacetyl-CoA molecule is then converted to HMG-CoA by the enzyme HMG-CoA synthase. This reaction requires a third acetyl-CoA, of which the CoA gets released. Then, the enzyme HMG-CoA lyase cleaves HMG-CoA to regenerate one acetyl-CoA and produce acetoacetate. Out of acetoacetate, the two other ketone bodies can be produced. Acetone is made out of the spontaneous decarboxylation of acetoacetate, and beta-hydroxybutyrate is made by the reversible reduction of one of the ketone groups with the aid of NADH. This reaction is catalyzed by beta-hydroxybutyrate dehydrogenase. Now, to understand the purpose of these molecules, it's first important to know that ketogenesis occurs in the mitochondria of liver cells. As we will see later in more detail, the liver is an anabolic organ that has the important role of making sure the rest of the body has all the nutrient it needs. Some organs like the brain rely almost exclusively on glucose to function. Consequently, when glucose levels go down, for example in conditions of starvation or fasting, ketone bodies will act as a metabolic fuel to shuttle acetyl-CoA across the body. To do so, the other organs that need energy from ketone bodies express the enzyme 3-ketoacyltransferase, which is able to take acetoacetate and convert it back to acetoacetyl-CoA. This reaction requires succinyl-CoA to donate its CoA, and in the process it becomes succinate. The acetoacetyl-CoA can then be cleaved to two acetyl-CoA molecules by thiolase. This allows to generate the energy that the missing glucose would typically provide in the tissues that need it the most. Obviously, since the mitochondrion of liver performs ketogenesis, it does not express the 3-ketoacyltransferase to make sure it does not consume the ketone bodies. So, to reiterate the purpose of this pathway and how it relates to beta oxidation, if someone is starving or fasting for extended periods of time to the point that the supply of glucose in the body depletes considerably, ketone bodies are a safety mechanism activated by the liver to supply that missing energy. Since beta oxidation can generate large amounts of acetyl-CoA, this is what supplies the starting material to make ketone bodies. Now, not all the ketone bodies are equal. Acetone is actually not used for energy as it gets released in the breath, and for beta-hydroxybutyrate, since it requires NADH to be made, its reconversion to acetyl-CoA, for example in a neuron, can generate back some additional NADH to make energy. So, in other words, beta-hydroxybutyrate offers the benefit of storing the energy of one NADH in the molecule. Alright, back to this overview of fat metabolism, we have now covered the catabolic pathways, namely the degradation of triacylglycerols to fatty acids and the degradation of fatty acids to acetyl-CoA via beta oxidation. We also discussed the production of ketone bodies and their importance when glucose is unavailable. Now, I want to consider the anabolic side of fats. The two main pathways for this are the synthesis of fatty acids from acetyl-CoA and the production of triglycerides from fatty acids. Thank you for watching this video. If there was anything unclear or there was a mistake somewhere in the video, make sure to let me know in the comment section. If you enjoyed this video and found it useful, you can consider leaving a like and subscribing to support the channel. Thank you again for watching, and I'll see you in our next discussion.